Hello, my name is Everardo Gonzalez, and I will present you the project of vaccine engineering in the time of COVID, rapid and cost-effective development of SARS-CoV-2 vaccine candidates in HEC293T cells and bacteria. And while the coronavirus possesses the largest genomes between 26 to 31 kb, and the main genes of this virus are present of 1AB spike, envelope membrane, and nucleocapsid. And you can see in this figure the SARS-CoV-2 genome and protein composition, where we have the non-structural proteins, the structural proteins, and the accessory proteins. The majority of these genome are composed with the non-structural protein. And in this map, you can see the situation of COVID-19, where we have more than 45 million of infection cases and more than 1 million of deaths in the world. And we have some vaccine strategies to treat this virus. We can use the whole SARS-CoV-2 inactivate, or maybe we can use only the RNA or produce DNA from the RNA virus. But also we can produce only the proteins of the virus or specific regions of action of a particular protein. And we are a Mexican research group in the University of Tecnológico Monterrey. We have the experience of production of recombinant proteins in bacteria and mammalian cells to treatment some infection disease. And here you can see the leaders and members from the Alvarez Trujillo lab. This slide is from our most recent work related with the Ebola, and it is important because it's the basis of this project. So with this vector, we can produce 2 or more proteins with the same promoter. And in this case, we produce a monoclonal antibody and a GFP protein with the idea to monitoring the culture during the time. In this slide, you can see the progression of the culture with the fluorescence in the cells and the characterization of the genetic and protein monoclonal antibodies. And here you can see another example of the technology that we can produce the GP protein from Ebola virus. And this GP protein is fused with RFP with the intention to see in the microscope and see the interaction between the monoclonal antibody produced in the previously and the interaction with anti-IgG market with FITC. In these figures, you can see the interaction. And here you can see the most important SARS-CoV-2 antigens, for example, the spike protein complete, composed by subunit S1 and S2, or maybe only the subunit S1, or the receptor binding domain. And the last one can be only the nucleoprotein. We have 2 platforms of expression. We produce in E. coli the receptor binding domain and nucleoprotein. And in mammalian cells, we can produce the subunit S1 and subunit S1 plus the virus domain called hold on. And well, this is the production workflow. We start with a culture transfected or transformed, depends on the protein that we want to produce. Then we follow by a lysis process or separation process to finalize a purification process using affinity chromatography. In each step, we use a characterization process using SDS page or Western blot to see the state of the protein. And here we have 3 different expression vectors to produce in E. coli. The first one is to produce the receptor binding domain, and the second and the third is to produce the nucleoprotein with variation of secretion or to produce in inclusion bodies. In this slide, we show the receptor binding domain that we can produce in E. coli. We can see the 3D model of the protein, and in a specific or receptor binding domain, we add two tags. The first one is a his tag to purify the protein, and the second one is the FH8 to increase the solubility of the protein. And well, this is important information related with the nucleoprotein from SARS-CoV-2. You can see the 3D model of the nucleoprotein, and we can produce this protein in E. coli. This is the genetic characterization from the E. coli clones. The first picture is the electrophoresis gel from the receptor binding protein. And the second picture is from the nucleoprotein clones. These amplicons are representative of the genes that codifies the proteins of the virus. And here we have the protein characterization. In the case of the receptor binding domain, we have a SDS page and also a Western blot using specific antibodies to recognize the receptor binding domain. And as a control, we use a commercial spike. And in the case of the nucleoprotein, we use a dot blot with serum from COVID-19 patient to recognize the production of the protein. And in the case of the mammalian cell production, we have 4 expression vectors to produce the subunit S1 and the subunit S1 plus the virus domain called foldon. This protein we will call F1 and S1 in the case of the subunit 1. And these 4 vectors have the possibility to produce the virus proteins and also produce the GFP protein to monitoring the culture. And well, the foldon comes from the fibritin of the T4 virus, and the idea of use this domain with the S1 of the SARS-CoV-2 is induce the trimer form that we can see in the nature of the virus and try to emulate the most similar the condition in the virus. We have previously works in Ebola virus, the formation of trimers with this foldon. So the idea is combine the foldon and S1 subunit to emulate this form in the virus. And here you can see the characterization of the vectors to produce S1 and F1 proteins in mammalian cells. And in this slide, you can see the protein characterization of the S1 and F1 proteins using a SDS page and a Western blot. You can see the supernatant and the cell lysate from the 2 cultures to produce the S1 and F1 proteins. And here we have the results of the transient production experiment of the F1 protein using puromycin to select the cells transfected and only media as a control. And you can see the culture of the 10 days that if we press with puromycin, we can produce around to 2.5 micrograms per milliliter of F1 protein. And here you can see the generation of stable pools from the S1 and F1 proteins. And this kind of cultures we can obtain using the antibiotic puromycin, do a pressure to the culture and kill all the untransfected cells. The idea is see the fluorescence that represent the expression of the protein of interest. In this case is the SARS-CoV-2 proteins. And well, with these antigenic proteins from SARS-CoV-2, we have some application. The first one, and well, we designed two formats of ELISA. The first one is a direct that are composed by adding the antigen produced by us, and as a control, we use a spike, a commercial spike, representing in red color. The RBD that we can produce in E. coli is represented with blue. And this is the first ELISA that we can design. And the second is a sandwich where we use the antihistamine antibody adding in the plate, and then follow the same steps that we can do in the direct ELISA. So, and you can see the results in the right side, respectively, of the direct and sandwich ELISA, and also similar between the commercial spike and the RBD that we can produce in E. coli cultures. And then we use this design of sandwich ELISA to test using sera from COVID-19 patients. And in this picture, you can see 50 samples where are detecting the specific antibodies from SARS-CoV-2. Here is important comment that we decide use a baseline with the support of samples that we have before of the pandemic time. So in this way, we are sure that we don't have the antibodies against the SARS-CoV-2. And the last application that we can do with these antigenic proteins is produce a vaccine candidate. So in this slide, we showed the experiment of animal model, and we have 4 groups of mice. The first group is composed by 3 mice that we injected 20 micrograms of receptor binding domain. The second group, we injected the same quantity of receptor binding domain, plus in the day 14, we inject 10 micrograms extra, like a boost. And the third group, we injected 20 micrograms of a conjugate of the receptor binding domain and nanoparticles that we produce in the Alvarez Trujillo lab. This conjugate is a new formulation of vaccine composed by nanofibers and the receptor binding domain. And the last group is the control, only the buffer where we resuspend the receptor binding domain. And in the right side, you can see the results of the specific antibodies from the blood from these animals. And you can see in the day 14 and day 28 that we can produce immune response with the different treatments. Be the best treatment the conjugate that I comment with the nanofibers and receptor binding domain. And well, the conclusion of this project, we can demonstrate a fast and cost-effective method to produce relevant SARS-CoV-2 recombinant proteins. We can produce 4 relevant antigenic proteins in E. coli, the receptor binding domain and the nucleoprotein. And in the case of the mammalian cells, HEC293T, the subunit S1 and the F1 protein that composed by subunit S1 and the foldon. And the SARS-CoV-2 antigens produced in bacteria and mammalian cells can be recognized by specific anti-SARS-CoV-2 antibodies. And well, I want to say thanks to FEMSA and to my university, Tecnológico de Monterrey. And thank you for your attention. And this is the information to contact the group. Thank you so much.